Hi class, welcome to the online session. We are going to discuss on 4 slides for gastric pathology, that's part 2. This would include 114, that's gastric lymphoma. We have slide 28 on acute appendicitis. We have slide 112 on amoebic colitis and 113 for pseudomembranous colitis. Okay, so let's start. I think you already have your manuals on for the second sem. Okay, so let's start. So our first slide would be on slide 28. This is acute appendicitis. Yes, acute appendicitis. If we are going to look at the slide, you would see a sagittal section or a cross section of the appendix, and it is dilated. You can see the central lumen has a lot of contents. We have here red blood cells, amorphous debris. I think these are food particles. Okay, and then you would also see a lot of inflammatory cells there. So you have segmenters. So 50 to 80% of acute appendicitis would be caused by obstruction. So it can be in the form of fecal material, a fecally, the presence of seeds, okay, or mucus that would be obstructing the lumen of the appendix going to the stool. So we have a dilated lumen. You can see the presence of an ulcer. This is part of the ulcer. And then you would see the presence of areas of necrosis. Sorry for the pink appearance of the slide. Okay, so you would see a lot of inflammatory cells. There's the presence of necrosis here. Do you remember the type of necrosis that we would see in acute appendicitis? Okay, so in this case, you would still see the presence of the glands. So you do not see the coagulative necrosis. Here, you would see the presence of inflammatory cells, segmenters, there. Okay. You can see the presence of segmenters. You can still see the presence of some of the muscle fibers, but in this case, there is none. So this is a form of liquefactive necrosis. It extends up to the periappendicial area. Okay, so that's periappendicitis. If we're going to look at the second slide that we have for acute appendicitis, notice that in this particular view, this is a scanning view. You do not see any luminal content, and the appearance of the glands here would be different. Okay. You can see that you would have ghost outlines of the glands here. So you have ghost outlines of the glands, but you can already see some of the cells to have absence of the nuclei or would have pycnosis or karyolysis. So this would already be coagulative necrosis combined with necrosis associated with inflammation, and this is an acute gangrenous appendicitis. Okay. And it extends up to the periappendicial area. So this is a periappendicitis. So next slide that we have would be 114. This is gastric lymphoma. Okay. So gastric lymphoma would fall under the marginal zone B cell type. Marginal zone B cell type. If you're going to look at the slide, we have the presence of aggregates of cells lying underneath the mucosa and muscularis mucosa. So we have submucosal collection of round cells. So how do you know that this is coming from the stomach. Okay, so always look for special cells. What do you call the cells? These eosinophilic cells. These are parietal cells. Okay? So you know that this is in the stomach. And then you look at this area. What is this? So we have two types of lymphomas based on the architectural pattern. It can be diffuse, it can be follicular. So do you see follicles here? There's none. So this is a diffuse type of lymphoma, non-Hodgkin's type. And then if you're going to look at the appearance of the cells, are they small? Are they large? So these are large cells. So usually in the histopath, we would call this one as a non-Hodgkin's lymphoma, diffuse, large cell type. And because it's in the stomach, there's a big chance that this is a marginal zone B-cell lymphoma. So marginal zone B-cell lymphoma would be a maltoma. When you say maltoma, it's a mucosa-associated lymphoid tissue tumor. And this can be associated with H. pylori infection. If we only have H. pylori and then we eradicated the H. pylori, there was the presence of the lymphoma accompanying it, it will also be eradicated. However, if there are certain mutations that would go along with it, like the translocation of 11 to 18 that would cause an API2-ML2 fusion gene, or the presence of another translocation causing the overexpression of MALT1, or another translocation causing the overexpression of BCL10, then these translocations will make H. pylori treatment ineffective for eradication of this particular lymphoma. Okay, so you need chemotherapy. So this is gastric lymphoma. So take note, usually the cells here, they have irregular nuclear membrane, okay, and then they have the presence of cleaving, okay, prominence of the nucleolus, and they are large cells. So this is gastric lymphoma. Next will be slide 112. Okay? Slide 112 is amoebic colitis. So amoebic colitis is caused by Entamoeba histolytica. Okay, so you have to differentiate this one from the next slide, which is slide 113. This is a colonic tissue. Okay, you can see the presence of those glands. Okay, an Entamoeba histolytica would show the presence of ulcers. Okay, the ulcers here would be flask-shaped. It means that the ulcer on top would be smaller, and then you have a neck, and then you have a large space at the bottom. It means that it can be in the submucosal or in the muscularis. Okay, so do we see the presence of a lot of nuclear debris or cellular debris, the presence of segmenters, okay? And then what do we have to look for? This one, okay? You see it? So this is a trophozoite. So an Entamoeba histolytica trophozoite would present with one nucleus, unlike that of the cyst wherein you see the presence of 4 karyosomes. Okay? So you have 4 nuclei. But in this case, you have 1 nucleus. How do you differentiate this far from a histolytica? Anyone? Okay, so you can differentiate them if you would see the presence of erythrocyte within the cytoplasm. So it means that it is it a histolytica or is it histolytica or this part? So you can see here the presence of the nucleus. So this one would be histolytica. Okay? So histolytica, if you would have the presence of erythrocyte. This is a trophozoite, another trophozoite. You have here the vacuole. Let's try to look for another one. There's another one, okay, with the nucleus. Okay, so I think you have a lot of examples already. So those are the things that you have to look for. And remember that the ulcer here would be lying underneath. Okay, it would be lying underneath underneath the under the epithelium. Okay, so this is amoebic colitis. Okay, our last slide for this session would be pseudomembranous colitis. So this is a type of colitis that is caused by an overgrowth of bacteria that is called the Clostridium difficile. So if we are going to give antibiotics, it will cause the eradication of the flora, and what's left behind would be the Clostridium difficile. So in essence, this is an antibiotic-associated colitis. And aside from that, the Clostridium difficile would release toxins A and B that would cause damage to the cytoskeleton. It will disrupt the epithelial lining. So again, if you would look at this slide, you would see the presence of colonic glands or intestinal glands. And then there's another ulcerative area, a lesion. Okay? So take note, this is lying above the colonic glands. So this is a pseudomembrane. Okay? So this entirety is a pseudomembrane. You would see the presence of amorphous debris. You have the presence of inflammatory cells, a lot of segmenters. Okay? And then you have to look at the characteristic appearance of the glands. Okay? Some of the glands appear to be distended. It contains a lot of necrotic debris and inflammatory cells. And then on top, it's open. Okay? In Robbins, it says that some of the glands appear to have an appearance of an erupted volcano. Okay? So this is it. That's the appearance of the glands that we would see in pseudomembranous colitis. Okay, so those are the slides that we have for this session. Okay, kindly go over with your manual. Kindly look into them and discuss among you what are the important answers. Okay, so thank you and have a nice day.